Microbes and Morgellons, Part 2. Chemical modifications to the C-terminus during transcription can also cause translation errors that might at least theoretically lead to a problem called prion disease. I discuss prion proteins in another section of this book. One kind of modification to the C-terminal end of the protein chain can take place with the addition of a phosphoglycan called glycosyl phosphatidyl inositol, or GPI. GPI is composed of fatty acids or glycolipids which attach to the C-terminus of the genome as a membrane anchor under certain conditions. Faulty GPI protein links are known to be responsible for the disease paroxysmal nocturnal hemoglobinuria, or PNH, also called Markia-Fava-Michelli syndrome, a rare autoimmune blood disorder that causes hemolytic anemia. GPI-linked proteins contain important peptide signals that direct them into the cell's endoplasmic reticulum. This is the inner part of the cell responsible for protein translation, protein folding, and protein transport to the cell membranes. Again, errors in protein folding might be at the root of prion and other diseases. As far as the science seems to reveal, prion diseases appear to be the most severe, worst-case scenario of adverse transcription errors. Prion diseases rapidly progress and are considered incurable at this point in time. Whether prion or other exogenous proteins are present within vaccines, human biologicals, and engineered biological controls that can cause such protein folding errors is entirely possible, especially when we know that scientists have released wild viruses and modified viruses that have been removed from their capsid as naked virus particles small enough to possibly be called prions. Some scientists have postulated that tiny microscopic species of Collembola or hexapods, which are a family of parasites that feed on leaf litter and decaying soil matter, may be implicated as a causative factor for Morgellons disease. But the evidence is that they may merely be accidental passengers to vectoring insect parasites. These creatures can be found in mold infestations, under leaky sinks, by swimming pools, or in the soil of potted plants. More importantly, they are commonly found in stored food products such as grains, and account for many losses in agricultural crops. Stored food pests are just as damaging and important to eradicate as pest insects on or within field crops, and biological controls are used against these pests regardless of their physical size. While some Collembola are flea-sized or larger, there are a few that are microscopic in size and too small to see with the naked eye. These creatures are part of the family of hexapods or springtails and are not considered to be true insects. Collembola feed on algae and bacteria, and some species are parasites of nematodes. So we can see how these organisms might be introduced into the bodies of Morgellons patients who are bitten by ticks or other insects that have been exposed to the nematodes used as biological controls. Ticks are parasitized by nematodes, and nematodes can certainly be parasitized by microscopic Collembola. If soil microbes or nematodes can ingest bacteria such as Agrobacterium tumefaciens, or polyhedrosis viruses such as Autographa californica, then we can certainly see how a disease like Morgellons may manifest in humans. Since nematodes were so widely applied as biological control agents, we can understand the nested pathogens that may lead to human infections once introduced by an arthropod vector, a perfect example of a microbial Matryoshka doll. Collembola are not considered by the CDC and others to be known human parasites. Regardless of whether Collembola are properly acknowledged by the public health sector, their accidental introduction into the human species through insects infested with Collembola-carrying nematodes may nevertheless facilitate human infections and, in effect, extend the normal host range for these pathogens. The idea that microscopic forms of this creature may be found in or on some skin lesions in patients with insect-borne infections should not be dismissed without proper scientific investigation by those who do not have ties to public health or industries related to agriculture and biological controls. According to a source, researchers from the Oklahoma State Department of Health and the National Pediculosis Association found collembola present on 18 out of 20 Morgellons patient skin lesions when examined microscopically. Perhaps the failure to acknowledge diseases such as Morgellons and their relationship to microscopic collembola has to do with concealing a relationship between the organisms and biological controls. If the collective we don't acknowledge fault or problems, then perhaps important public health issues can simply be ignored. I suppose to some it is simply easier to call patients delusional than it is to address important agents that seem to be making thousands of patients the world over incredibly ill. We already know that Bt toxins and other genes can be introduced into plants using vectors such as agrobacterium, in combination with a baculovirus vector such as Autographa californica to create biological controls. The question to ask is, why won't scientists and public health agencies who have known about biological controls all these years acknowledge the presence of these pathogens and the idea that they may be causing human diseases? Again, what happens after insect transmission of these foreign proteins into humans may be largely determined by the menu of resident viruses and toxins within our bodies and the bodies of the vectors. Also playing a probable role in disease is the ability of the engineered chimera and their toxins to degrade our gut, enter our system, and activate latent infections. Other factors that determine final disease presentation include our genetic makeup, chemical and heavy metal exposures in us or the vector as nested pathogens, and any transcription promoters which may be present or to which we may be exposed at a future date. It is these unpredictable elements that I fear are initiating some of the world's most common disease epidemics of today, such as Lyme, RA, MS, ALS, or Morgellons, or even some that to date have, quote, no known cause, end quote. If so, and if scientists, public health experts, and industry partners are aware of this information, and they seem to be, would this knowledge provide a strong reason for patients to be diagnosed with delusional parasitosis? Or, as in the case of Lyme disease, patients to be told it's all in your head? If illnesses have their origins fundamentally within the agrobiotech industries, might we find many eager to silence patient populations in favor of the pursuit of product pipelines and concealment for liability purposes? The revelation of such connections might necessitate costly changes to the highly profitable agrobiotech pharmaceutical industries. If so, wouldn't the prudent thing be to investigate, admit defeat, and go back to the scientific drawing board to create products that are both effective and safe? And wouldn't it be far better to acknowledge the patients who suffer from diseases than to simply attempt to sweep them under a grand carpet of denial? At some point, the lump under the carpet will grow too large to ignore and will require audible acknowledgement. Regardless of a patient's creative imagination, it is impossible to manifest identical physical symptoms in thousands of unrelated people. Therefore, those who choose to ignore what has become painfully obvious to thousands around the world may be suffering from a nihilistic delusion of their own.